Welcome, everyone, to Episode 2, The Chemicals of Life, Remastered. Today, I'm going to explore the origins of the biological molecules that gave way to the first replicating cells. This includes the chemical elements of life, the all-important water, and the chemical evolution of the early biomolecules. Let's start out at the very beginning, going 4 billion years back in time to the late Hadean. Here, on the young and primordial Earth, life is thought to have been first generated in the biomolecule-rich muck that covered the surface. The Hadean Eon began with the slow formation of the Earth, nearly 4.6 billion years ago. For hundreds of millions of years, the molten Earth accumulated the mass of its orbital neighbors. Lava flows swept over the plains of the first proto-continents as tectonic plates formed and cracked and ground past one another. As the Earth cleared its orbit, meteors would rain from the sky in a terrifying perpetual storm of flaming rock. At some point, a planetary body known as Theia collided with the Earth. Theia is predicted to be roughly the size of Mars, so the impact would have been incredible. It created a so-called ocean of magma on the surface, throwing an immense layer of vaporized rock into the atmosphere, and it threw a debris field out into space. This debris field was about 20% of Theia's mass. Some of it would fall to Earth, and some of it would fly off into space, lost forever. But about 50% of this debris field would eventually coalesce to form the Moon. The layer of rock vapor in the atmosphere settled within a couple thousand years, while heavier material in the boiling oceans sank into the Earth's mantle. The Earth's atmosphere now is a hellish 446 degrees Fahrenheit, choked with carbon dioxide and nitrogen, and very little, if any, oxygen. Volcanoes are common, erupting at up to 100 times the rate that they do today. There was so much carbon dioxide that the atmospheric pressure was 27 times what it is today. This is the equivalent of being 270 meters under the surface of the ocean, where the pressure is absolutely immense. In order to survive in these conditions, you would need a really hardy suit that can protect you against high heat and pressure, and you would need a specialized mixture of gases to breathe so that you don't get nitrogen narcosis or carbon dioxide poisoning. This carbon dioxide was slowly absorbed into the oceans and sucked into the mantle through the actions of plate tectonics. The oceans themselves were hot, roiling pools, rich with sediment and minerals and the chemical building blocks of life. 4 billion years ago, the Hadean Eon gave way to the Archean Eon. This new time period saw the gradual cooling of the Earth. The planet's orbit was largely cleared, so asteroid impacts were a lot less common. Plate tectonics were slowing down, and they were stabilizing the geography of the landscape, and atmospheric heat slowly declined. Now, even though things were calming and cooling, things were still pretty rough. The landscape was still changing, just at a slowing rate, and the atmosphere was still pretty hot relative to today. Now, it's around this time, the late Hadean and the early Archean, that life is believed to have first appeared. It's difficult to precisely identify the first life form, but we have identified life that existed as early as 3.7 billion years ago, in the form of biogenic graphite found in Greenland, or 3.48 billion years ago in the fossils of microbial mats found in Australia. But how did these microbes first come about? What was the environment that enabled their genesis? And what are they made of? Living matter incorporates a number of elements, the primary four among them being hydrogen, carbon, oxygen, and nitrogen. Carbon is in all living things, such that one of the defining characteristics of a biomolecule is that it has a core structure composed of carbon. Now, what about carbon and all of these other elements makes them so critical for life? To understand this, we have to examine these elements and the organic compounds that they form on an atomic level. A very quick recap on basic atomic chemistry. The atom is the smallest particle in chemical organization that retains distinguishing qualia. The atom has two parts: an extremely tiny, dense nucleus of protons and neutrons, and an electron cloud with one or more electrons. Now there's different kinds of molecules, and there's different kinds of atoms, but protons, neutrons, and electrons are all the same, no matter which particle they're in. The atomic weight of an atom is the summed mass of these parts, measured in units called daltons. Neutrons and protons both weigh about 1 dalton, while electrons are so small that their mass is generally ignored. The atomic number is the number of protons that exist in the nucleus. The number of neutrons in the nucleus determines the isotope, or alternative atomic structure of a particular element. The word isotope means equal places, referencing their position on the atomic table. Carbon-12, for example, has 6 protons and 6 neutrons in its nucleus, while carbon-13, which accounts for about 1.1% of the planet's naturally occurring carbon, has 6 protons and 7 neutrons. Both carbon-12 and carbon-13 are stable. They're both stable carbon isotopes, but carbon-14 is not stable. Carbon-14 has 6 protons, like all carbon atoms, but it has 8 neutrons. And this arrangement is unstable, making carbon-14 a radioactive isotope. The decay process of carbon-14 sees a neutron convert into a proton, gaining a positive charge, and turning the atom into a stable nitrogen isotope, with 7 protons and 7 neutrons. So, where is the electron in all of this? The electron exists in a field, orbiting around the atom called, unsurprisingly, the electron field. Now, the electron is not literally orbiting in the same way that moons orbit planets. It's often used as an educational device to illustrate the atom as billiard balls, and the electron is a smaller billiard ball just orbiting around the nucleus, which is a slightly larger billiard ball. This isn't really how it is. Instead, the electron exists in one of multiple specific regions called orbitals. The orbitals are lump-shaped, three-dimensional volumes that surround and extend from the nucleus in layers of geometric patterns that overlap, called electron shells. The larger the nucleus of an atom, the more electrons that it has. As each orbital can only hold 2 electrons, atoms that have many electrons will have many more filled orbitals, with the innermost shells filled first. For example, a shell that has 4 orbitals can hold 8 electrons. The three-dimensional shape of these filled electron shells, these electron clouds, is a really fundamental and important aspect of the atom's geometry, and it dictates how the atom interacts and bonds with other atoms, and it explains why organic molecules take the shapes and the conformations that they do. Carbon dioxide, for example, is a linear molecule. Each oxygen atom sits on opposite sides of a carbon atom, forming a straight line. This straight line is due in part to the double bonds that either oxygen atom has with the carbon atom. Now, if we look at water, or H2O, is a planar molecule, meaning that it's not linear, but it exists in a single plane. So why is water planar and not linear? Well, in a water molecule, oxygen is bound by 2 hydrogen atoms, but it also has 2 groups of paired electrons that aren't shared with any other atoms. Now, all 4 groups of the oxygen's electrons, the 2 pairs that are just unshared around the oxygen atom, and the 2 pairs that are being shared between the oxygen and the hydrogen atoms, all 4 of these groups are negatively charged, and so they repulse one another. To see the geometry of the water molecule, touch your thumb and forefinger together, such that the circle your fingers make is parallel to the ground. Now with your other hand, do the same, but have the circle that your fingers make be perpendicular to the ground. Now if you touch all 4 of your fingertips together, at the center of this shape would be the atomic nucleus. The tips of your fingers represent the direction that the orbitals of this particular shell are organized. The 2 electron pairs sit in one plane of the oxygen atom's orbitals, while the 2 shared electron pairs sit in the other plane of the oxygen atom's orbitals. This arrangement makes the water molecule appear bent, like a boomerang or the letter V, while remaining otherwise flat. Now the molecule methane, on the other hand, is tetrahedral. Now imagine the same shape that your fingers made earlier with the water molecule, except this time the center of your fingertips is a carbon atom, and each orbital is filled with the shared electrons of a bound hydrogen atom. The filled orbitals repulse one another, so they all point away from each other. They try to point as far away from each other as they can. And so this means that as they point away from the nucleus, they form a tetrahedral shape. In all of these examples, the electrons that are being shared are part of the outermost shell of the atom, the valence shell. To keep things nice and simple, the electrons in the valence shell are called valence electrons. An atom's valence is the number of these valence electrons that it has. Now carbon has 4 valence electrons, all of them unpaired, which allows it to bind up to 4 other atoms at a time. This versatility and modularity is the primary reason why carbon is so fundamental to life. It can bind with damn near anything. It can make sheets or fibers or clumps or whatever else. Now oxygen has 6 valence electrons, but only 2 of these are unpaired. As I mentioned earlier, oxygen has 2 groups of paired electrons. The unpaired valence electrons in oxygen and in carbon and in every other atom, these are the electrons that are involved in covalent bonding between 2 atoms. The covalent bond is just one of several types of bonds. The covalent bond involves 2 atoms, each sharing an electron from their valence shells. Both shared electrons form a pair, and this shared pair binds the atoms together. This is called a single covalent bond, where you have 2 shared electrons, one donated by each atom. But there are also double bonds, which exist when there are 2 pairs of shared electrons between 2 atoms. These bonds are stronger, they hold the atoms closer together, and they require more energy to break than a single bond. Each oxygen atom in the carbon dioxide molecule is bound to the central carbon with a double bond. This central carbon has 4 valence electrons, so it's able to donate 2 of them to each oxygen atom and engage in this double bonding. Now as each oxygen atom has 2 valence electrons, both of those are shared in the double bond with the carbon's electrons. Now further still, there's the triple bond, where 3 pairs of electrons are shared. Triple bonds are even stronger than double bonds, and they hold each atom even closer together. A common example of a triple bond exists in molecular nitrogen, N2. Each nitrogen atom has 3 valence electrons. So 2 nitrogen atoms can then bind together and each share their 3 valence electrons to form a triple bond. These covalent bonds can be nonpolar, where the electrons are shared equally between the atoms. Or covalent bonds can be polar, where the electrons are concentrated closer to the more electronegative atom. Electronegativity is a property of atoms determined by the number of protons in the nucleus, and the distance between the valence shell and the nucleus. Now, I kind of like to think of electronegativity and the electrons through a kind of gravity metaphor. You can think of the nucleus as something like the center of mass of a planet, which determines its total gravity signature in space. The surface gravity is dependent upon both the total mass of the planet and the radius of the planet, or the distance between the center and the surface. Now the valence shell works similarly. The valence shell is like the surface of the planet. The closer it is to the center of mass, to the nucleus, the stronger the surface gravity, the stronger the electronegativity. With an atom like oxygen, you have 8 protons, but only 2 electron shells, and this makes oxygen very electronegative. There are other heavier atoms that have more protons, but they also have more electrons, and so they have more electron shells. As the valence shell is far from the nucleus, the influence of the atom's electronegativity on other atoms it binds with is lessened. Now here's a really cool thing about the periodic table of elements. If you start at the top of the table and you go from left to right, the elements have an increasingly strong electronegativity as the number of protons in the nucleus increases. The elements in the far right column have full valence shells, so they don't readily participate in covalent bonding. They don't really have an unpaired electron to share. Now, as you go down a given column on the periodic table, the electronegativity decreases as the increased number of electron shells pushes the valence shell farther and farther away from the nucleus. Nonpolar bonds can occur between atoms that have a weak electronegativity, or a similar electronegativity. For example, you have the bonds between carbon and hydrogen. These are relatively nonpolar. But then, on the opposite end of the spectrum, you have polar bonds. Polar bonds occur between two atoms that have a distinct difference in their electronegativity. Where a carbon-hydrogen bond is nonpolar, an oxygen-hydrogen bond is polar. The more electronegative atom, in this case oxygen, pulls the electrons closer to itself, and because the electrons have a negative charge, this concentrates a partial negative charge around the electronegative atom, and it creates a partial positive charge on the other atom. So in a water molecule, where you have H2O, the oxygen atom has a partial negative charge, while both hydrogen atoms have a partial positive charge. This makes water a polar molecule, which is really important, but I'll get into this in more detail in a few minutes. When these atomic particles are near one another, they begin to organize themselves in a three-dimensional lattice, according to these partial charges. The partial positive charge of a hydrogen atom is attracted to the partial negative charge of the oxygen atom of a nearby water molecule, and this is a fundamental example of a second type of bond, called a hydrogen bond. If the hydrogen atom is bound to an electronegative atom, like oxygen or nitrogen, the electronegative atom will have a partial negative charge, and this hydrogen will subsequently have a partial positive charge. So all of these partially positively charged hydrogens will be attracted to partially negatively charged atoms floating around, and they'll loosely try and bind to them. This is the hydrogen bond. Because it's kind of a loose connection, it's much easier to break than a covalent bond, because electrons aren't being shared. It's just a polarity attraction between positive and negative charges. If you consider our DNA, you'll see that it's built of 2 sugar phosphate backbones, each lined with nucleotides. In the DNA helix, these nucleotides interlock with each other, with 2 or 3 hydrogen bonds. To read the DNA, an enzyme called DNA helicase has to unzip the DNA helix by breaking these hydrogen bonds. A third type of chemical bond, besides the covalent and hydrogen bonds, is the ionic bond. Ionic bonds do share electrons, as happens in the covalent bond. However, in an ionic bond, the electronegativities of each atom have such a huge difference between them that one electron is entirely transplanted from one atom to another. There's no sharing. It's purely, here's an electron, I'm handing it over to you. These ionic bonds typically occur between atoms that have 1 electron in their valence shell and atoms that need 1 more electron to fill their valence shell. The lone electron will leave its host atom, knocking its valence shell down to a filled level, and the outgoing electron joins the receptor atom, filling its valence shell and stabilizing it. Both atoms are made more stable by this ionic bond. An example of this in our daily life is table salt. Table salt is composed of sodium and chloride in an ionic bond. The lone valence electron on sodium's outer shell leaves and takes its negative charge with it. This turns sodium into a cation, a positively charged ion. The electron and its negative charge will come into the electron field of the chloride atom, joining its 7 valence electrons to fill out its valence shell. This makes the chloride atom an anion, or a negatively charged ion. All of these ions and other atoms and molecules are all floating around in a solution. They're colliding with one another, bouncing off of each other, hitting each other and other things. They're binding to this, having bonds broken with that. It's really chaotic. It's a mess. This solution that everything is floating around in, as you might have guessed, is water. Now, as I mentioned earlier, water is both polar and it has a bent orientation, which gives the molecule a very versatile 3-dimensional polarity. Polar molecules and ions, basically any particles that have a charge, will interact in some way with the partially charged poles of the water molecule. Each pair of partially positively charged hydrogen atoms can use hydrogen bonds with almost any other charged particles to make them dissolve in water. Consider salt again. When you put salt into water, the salt dissolves. The negatively charged chloride atom attracts the positively charged hydrogen tips of the water molecules, while the positively charged sodium atom attracts the negatively charged oxygen of the water molecules. Molecules of water will thus surround each atom and break their ionic bond, with each ion then going in whichever direction in the watery solution. This is what it means for something to be dissolved in a solution. When a substance is able to be dissolved in water in this way, it's hydrophilic, meaning water-loving. This is, of course, as opposed to substances that are hydrophobic, or water-fearing, which don't interact with water through hydrogen bonds. Consider a long chain of carbon. All the carbon is linked together, and every other valence electron is paired with a hydrogen atom. This hydrogen-saturated carbon chain has no strongly electronegative atoms, so it doesn't have a charge. It's a nonpolar molecule. There is no electronegative atom for a water molecule to form a hydrogen bond with, because there's no partially negatively charged atoms and no partially positively charged atoms. And so water just can't interact with the carbon chain via hydrophilic reactions. In other words, this carbon chain is hydrophobic, and in a water solution, it'll react with other hydrophobic molecules and clump together to reduce the surface area in contact with the polar water molecules. The polar nature of water gives it a lot of really cool properties, and it makes for a really unique solvent. Perhaps the most famous property of water is the fact that it's denser as a liquid than it is as a solid, which is a sciencey way of pointing out that ice floats. When water freezes into its solid state, ice, the water molecules crystallize into a 3-dimensional lattice arranged with hydrogen bonds. The lattice structure has a lot of open space between molecules, due to the honeycomb skeleton pattern that emerges. When you add a little heat to the system, these hydrogen bonds begin to break, and the ice turns to water. The fact that ice is less dense than water is critically important. Because ice floats, it forms a shield over any given body of water. This ice shield on the surface of the water helps regulate the water's temperature by trapping heat in and keeping cold air from touching the surface. Now, if it was the other way around, if ice sank, the ice wouldn't get any heat from the sun, and it wouldn't melt. Instead, it would gradually cool all of the water in its proximity, and this would eventually freeze out the entire body of water. If this is how water acted, life would not be possible. Now, another cool property of water is surface tension. Surface tension is a result of the cohesion that exists between water molecules. Due to the huge branching chains of water molecules that are all connected with hydrogen bonds, water exhibits a strong cohesion. This is why you can fill a cup slightly more than full, and the water will round off on the edges as it makes a slightly bubbled top. This is caused by the countless trillions of hydrogen bonds going on between all of the water molecules. Now, for the same reason, water is also adhesive, as it sticks to the polar surfaces that it touches, again because of hydrogen bonds. Hydrogen bonds are why, after something like a cup or a skillet has water splashed on it, small drops of water will stick to the surface, instead of just running off the side. Hydrogen bonds are also why it's difficult to move through water quickly. The resistance that you feel while swimming underwater, or just swimming through the water in general, is due to the collective trillions and trillions and trillions of these hydrogen bonds that you're trying to push through. Now, water also has a very high specific heat, or the amount of energy that's needed to raise the temperature of 1 gram of the substance by 1 degree Celsius. Because of the huge number of hydrogen bonds that need to be broken, it takes a pretty considerable amount of energy to heat up water. Similarly, water has a very high heat of vaporization, or the amount of energy that it takes to turn that same gram of substance from a liquid state into a gaseous state. When sweat evaporates off of your skin, it takes with it a little bit of the heat that you generate while working out or doing strenuous activity. By evaporating and taking with it a little bit of heat, sweating cools you down. Not only does water have all of these great properties, it's not even a fully stable molecule. At any given time, some portion of the water molecules in your water bottle are dissociating into OH-, a hydroxide ion, and H+, a hydrogen ion. The hydroxide ion, the negatively charged OH molecule, is a base. The hydrogen ion associates with another water molecule, its positive charge attracted to the partial negative charge of this other water molecule's oxygen atom. This will create H3O+, a hydronium ion, which is a positively charged water molecule with 3 hydrogen atoms. This hydronium ion is an acid. So what makes the hydroxide ion a base, and the hydronium ion an acid? During a chemical reaction, acids are chemicals that donate a proton to whatever else they're reacting with, just like the hydrogen ion that's produced by the dissociation of water. The hydronium ion is an acid because in a chemical reaction, it will donate that third hydrogen atom to return to a more stable form. Water is a pretty weak acid. The stronger the acid, the easier it is for the molecule to donate a proton. And water, or these hydronium ions, they're acidic, but they're not particularly strong. Now, bases are chemicals that accept those protons, like the hydroxide ion, which has a negative charge and a strong affinity for the positive charge of any proximate hydrogen ions that are floating around. When you add an acid to a solution, you're basically increasing the concentration of hydrogen ions or protons in that solution. Now, if you add a base, you will lower the concentration of these free-floating protons, because some of them will get accepted into the molecular structure of the base. The pH scale is a logarithmic measure of the concentration of protons in a solution, where 0 is the most acidic, and 14 is the most basic, with pure water sitting at a neutral pH of 7. Although if you have water sitting out exposed to the air, the water will react with carbon dioxide in the atmosphere to form carbonic acid, and this will slightly decrease the pH of the water, so somewhere in the mid-6s. It should also be said that 0 and 14 aren't the maximum end caps of the pH scale. There's some superacids and superbases that go beyond this, but I don't understand the chemistry of those crazy substances, so I won't talk about them in too much detail. Anyway, the pH can be determined mathematically, being defined as the negative log of the concentration of protons in the solution. Now, inside of a cell, the pH of the water has to be controlled to maintain proper functioning of the organelles and the internal enzymes. This maintained internal condition is called homeostasis, and it applies not just to cells, but to entire organisms as well. Your body uses mechanisms to regulate your internal temperature, just as buffers regulate the internal pH of your cells, minimizing the effect of an added acid or a base. Alright, so now that I've discussed the elements, I've discussed bonding, and I've discussed how awesome water is, it's time to address the next big question. How did these starting materials come together to create the first biomolecules and the first cells? In essence, how did the last universal ancestor of all life come about? Some major competing hypotheses are the surface metabolism model, which theorizes that organic molecules formed when gases interacted with the mineral surface of deep-sea vents. And there's also the prebiotic soup model, which theorizes that organic molecules appeared in gases in the atmosphere and collected in the ocean through rain. There have been a number of really interesting experiments that have recreated the primordial conditions of the early Earth to see if they can generate biomolecules. Perhaps the most pioneering and famous example is the Miller-Urey experiment that was initiated in 1953. Stanley Miller was a graduate student and teaching assistant at the University of Chicago. Due to his position on the campus, Miller attended numerous seminars and speeches. Now, do you remember the Hadean Eon that I discussed at the beginning of the episode? The physical chemist Harold Urey was giving a lecture on the Hadean era and the conditions of the early Earth, which captivated and intrigued Miller. Miller met with Urey in 1952 to discuss his experimental plans for recreating the conditions of the primordial Earth. At first, Urey was hesitant, but Miller suggested that they test electric discharges to see if that could encourage the formation of biomolecules. After all, the early Earth was characterized by terrible storms and frequent lightning encouraged by the heavy volcanic activity. The experiment used a large glass flask filled with methane, hydrogen, and ammonia to simulate the atmosphere of the primordial Earth. 2 electrodes sat within the large flask, reaching into the atmosphere to induce sparking discharges of electricity. There was 2 tubes that connected the large flask to a smaller 200 milliliter flask that was filled with water, which represented an ocean and which was continuously boiled. The boiled water produced steam, which was cycled into the atmosphere flask. Here the steam cooled, and it would drip back into the ocean flask through a second tube. This produced a circulatory effect, where any biomolecules produced in the atmosphere flask would be flushed down into the ocean flask. With the water circulation in place, they began to regularly activate the electrodes, zapping the mixture of gases in the atmosphere with pulses of electricity. These electric pulses affected the gases in the flask in much the same way that a lightning bolt would have affected the gases in the atmosphere of the early Earth. Within a day of regularly sparking these electrodes, the water in the ocean flask had turned pink with biomolecules. By the end of a week of regular sparking, the ocean flask was a deeply opaque red, packed with formaldehyde, acetic acid, hydrogen cyanide, amino acids, nucleic acids, and all sorts of other complex organic compounds. These organic compounds are composed of a carbon-hydrogen backbone, or skeleton structure, with side groups that contain hydrogen, nitrogen, oxygen, sulfur, and phosphorus. These side groups are called functional groups because they add qualities to the molecule that allow it to participate in or carry out certain functions. There's 6 basic functional groups that are typically found in the carbon backbone of simple organic molecules. These are the amino group, the carboxyl group, the carbonyl group, the hydroxyl group, the phosphate group, and the sulfhydryl group. So to start at the beginning, the amino group is a base characterized by a nitrogen atom with 2 hydrogen atoms attached to it. The nitrogen atom has a partial negative charge, which attracts the positively charged hydrogen ions given off by random nearby acids. Carboxyl groups are one such acid, consisting of a carbon atom attached to 2 oxygen atoms. One of these oxygens shares a double bond with the carbon, while the other oxygen is bound to both the carbon and a hydrogen. The carboxyl group is an acid, because that hydrogen is donated in a reaction. Amino acids are a really important class of biomolecules that compose all of the proteins in your body. and they have both an amino group and a carboxyl group. These two groups can be joined in a covalent bond, which allows the amino acids to be linked up to form a long polypeptide chain. The carbonyl group is similar to the carboxyl group, except there's only one oxygen atom attached to the carbon. The carbon still has the double bond with the oxygen, but it shares its last electron with either a hydrogen or another carbon. If the carbon of a carbonyl is bound to a hydrogen, that carbonyl is called an aldehyde. If the carbon is instead bound to another carbon atom, presumably part of some larger carbon chain, then that carbonyl is called a ketone. The hydroxyl group are highly polar alcohols, consisting of an oxygen atom bound to a hydrogen. The high electronegativity of the oxygen atom makes the hydrogen partially positively charged, encouraging the formation of hydrogen bonds that make the hydroxyl group react with other polar molecules, and it also encourages the chemical's solubility in water. The phosphate group is a large side group that consists of a single phosphate atom that has 5 bonds to 4 oxygen atoms. One of these oxygen atoms enjoys a double bond with the phosphate atom. Another oxygen is bound to both the phosphate and a carbon atom in the carbon backbone. The remaining 2 oxygen atoms have only 1 bond each with the phosphate, but they lack any bonds with hydrogen atoms. This means that the remaining 2 oxygens are negatively charged. As this gives the phosphate group 2 negative charges, it's a pretty strong base. It's pretty strongly attracted to any floating protons zipping around the solution. Now the sheer quantity of oxygen here, 4 atoms, it makes the phosphate group very electronegative. Because it's so electronegative, it has a very profound effect on all the other electronegative atoms in a molecule. It can push them around or pull them towards it. And so if you transfer a phosphate group from one molecule to another during a chemical reaction, for example, it can often cause significant conformational changes in both compounds. This particular detail of biochemistry is integral to a lot of transport proteins that use energy to move ions and other particles across a membrane. Lastly, we have sulfhydryl groups, which are similar to hydroxyl groups, except that a sulfhydryl group possesses a sulfur atom instead of an oxygen atom. Sulfhydryl groups have a very interesting and a very useful property. When the hydrogens on the end of 2 groups are removed, the 2 exposed sulfurs can bond together to form a very strong disulfide bond. These disulfide bonds are like very sturdy staples among the soft clumps and fibers of organic material, and so they offer a degree of mechanical stability, and they're really integral to maintaining the specific shape and structure of particular biomolecules, like proteins. These functional groups are just some of many other molecular building blocks that are part of a huge pool of different molecules that all underwent chemical evolution. The most sophisticated proteins, lipids, amino and nucleic acids, carbohydrates, and self-reproducing RNA, the building blocks of all living things, slowly appeared as products of this chemical evolution. RNA alone possessed the ability to self-replicate itself, and the first RNA can be argued to be the first true baby steps of life. As the RNA got more sophisticated, it recruited proteins and carbohydrates to assist it in its replication. It became more complex, and eventually became double-stranded, forming DNA. Now when you have lipids, like phospholipids, when you dump a bunch of these in a polar solution, like water, these phospholipids will spontaneously form membranes. They'll spontaneously form giant bubbles, which are really stable structures because they totally minimize the contact between polar water molecules and the hydrophobic regions of the phospholipid molecules. When you have RNA or DNA and all of its associated proteins and carbohydrates all floating around within one of these membranes, you have the first cells. I hope you enjoyed this episode of the podcast, and I hope you learned something cool. I hope there was something neat here that you could take away from this. In the next few episodes, I'll explore the various groups of biomolecules that emerge from this primordial ooze, beginning first with amino acids and proteins in episode 3. Thanks for listening.
Would you like to support the Biologic Podcast? It's super easy. When you open a new episode, press the like button, share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.